Okay, well, good afternoon, everybody, and thank you for coming to another one of our TESS seminars. As you know, TESS is our Center for Tropical, Environmental, and Sustainability Science, which is one of our main terrestrial ecology forums here at JCU. It's a real pleasure. I wanted to mention also, this is the last semester, seminar of this semester, okay? So that finishes up here. And then Annabelle, do you know offhand, we have a few weeks off, how much time?
Yeah, I think we start getting around about 22 months.
Okay, well, so that's about 2 months, is that right? Something like that? Yeah, we have quite a little break in there. But anyway, we'll be seeing you all before too long after that. It's a real pleasure to introduce Renee Catullo, and apologies, Renee, that we've sort of kept you waiting here. We're sort of transitioning to this online thing, and a lot of our audience is now watching our talks, but it's online, and we've had up to 150 people in a seminar, but huge numbers of them are following things online. So it makes it a little bit different for us and unusual for us. Anyway, Renee has done a lot of interesting work. She got her did her undergraduate studies at Oregon State University. Renee, you're Aussie, right? But you went to the US for your undergraduate work?
No, I came here for an Australian.
Oh, okay. Sorry, I didn't pick up the accent. Yeah. Okay. Well, anyway, so you were from the U.S. originally.
Yeah.
Okay. I grew up just a stone's throw from Corvallis. I was always going to go to Oregon State and be a marine biologist. That was my aspiration in life. Didn't quite happen, but anyway. So Renee studied there. She studied at ANU, and she's also been at the University of Western Australia for a number of years or several years. She's interested in amphibians, and she's interested in conservation biology, conservation genetics, and she uses conservation genetics as a tool to study endangered wildlife and endangered ecosystems like those arising from the Black Saturday fires. So without any further ado, we'll thank Renee for coming and joining us, and really appreciate it, Renee. Thank you.
Thank you. I'll get started. So, yes, I have a big interest in genetic diversity, and well, if you need me to be a biologist out in the field doing something, it needs to be frog related. I do work more recently extremely broadly across taxonomic groups. I thought I was getting widespread moving into mammals, but at this point we're also moving into reptiles, plants, various organisms. And really that's kind of sparking that broader interest, which kind of extends from work I did as a postdoc with Simon Ferrier at CSIRO on thinking really top down about ecosystems and what does that mean for planning. And so, we do these loops away from sort of our early starts and now coming kind of full circle to really wanting to think about that spatial aspect of planning. So first of all, we'll just do a first year biology slide. Why is genetic diversity important? So genetic diversity, it provides that understanding of the variation in genes in a species, in a population across individuals. And one way it's very much used is as a proxy for adaptive potential, particularly within the context of landscape change. We've got a sort of classic paper here by Carlos Segro at Monash University. And it's used as a proxy for species resilience to change and their evolutionary potential. So how well are they going to be able to adapt, with low diversity species being expected to adapt more poorly and have greater risk of extinction? Now, there's lots of arguments about whether neutral genetic diversity and adaptive potential or adaptive genes are really linked, but most of the arguments against it, while containing some valid points, also contain ways around it that are physically impossible for most species. So it involve physiology experiments, all sorts of things that just would not be feasible. So at this stage, my view is genetic diversity is still our best proxy for adaptive potential for the vast majority of the diversity on the planet. And we also know that outside of adaptation, it's really important that we maintain diversity so we can avoid the negative effects of diversity loss. So inbreeding load, which is a reduction in fitness because of having two copies of an allele because of the relatedness of your parents, and those are often bad for you. And genetic drift, which basically means you lose really important alleles through stochastic factors and works against selection. So your adaptive potential is reduced just because you're losing really important variation. And just thought I'd throw a classic here, sort of an Allendorf book out on conservation genetics and the genomics of populations. And he really outlines the kind of practical applications of conservation genetics. And I really work across these three in bold, which is the management of populations and biological communities, and then describing population structure, relationships across different species and taxonomic groups, and understanding how effects of sort of anthropogenic change, or how anthropogenic change affects species genetic diversity. So I just thought I'd throw in a single recent paper. This is not the most exceptional population genetics paper in the world, but it's mine and it's good. So I thought I'd do it in my talk. So this is a recent paper by honors student Rhiannon and red-tailed phascogales, which are essentially adorable little antechinus with floofy tails. This particular species used to be distributed really widely across Australia, and it has now retracted to basically where these white dots are, which are these reserves. So this is sort of the entire distribution of the species, and this is actually a reestablished translocation. So they persist in this, despite being an arboreal species, persist in this highly fragmented landscape. And there's a lot of questions. So this phascogale is probably mostly impacted by cats. And in predator-proof reserves, they actually go gangbusters. So as soon as you get rid of cats, these guys are doing great. That's the main threatening process. So we did a bunch of genetic work on this. So essentially, this was just one of the results. But what we have here is a structure plot where each bar represents an individual, and the colors represent that individual's relative proportion of an ancestral number of populations. And when you see a graph that's basically like this, that just basically suggests isolation by distance. And indeed, that's what we found. This is one of our kind of best news stories for a federally vulnerable listed mammal you could get, which was actually, we didn't see a lot of heavy population structure in this. So our reserves don't seem to be particularly isolated. So these little guys do seem to be moving around a lot. And this map is probably the most important one. Essentially, it's like a topographic map where the width of the bars represents a gradient. And if your bars are pretty evenly spaced, then that's your transition in basically genetic distance between populations. And since we have really evenly spaced bars, these guys move around. They seem to be able to go 10, 11 kilometers without too much problem. So this is just sort of a classic study. We were able to make management recommendations. So they clearly intermix, so manage them all as a single unit. We didn't find significant signs of inbreeding or drift, but we did find slightly higher relatedness within reserves than between reserves. And so all of this kind of told us we do need to maintain and improve habitat corridors between each of these little reserve locations. And these two northernmost reserves were a bit more isolated than others. And so improving that connectivity was really important. And if you want to go take a bunch of phascogales and put them in a predator-proof reserve, your best bet is to drive in a big circle and grab an individual or two from every reserve and chuck them in a predator-proof fence, and you're going to get your most diverse, unrelated population you can get. But the reason I'm showing this is not really about phascogales, but about the fact that we go out and we sample all of these populations and we generate all of these metrics that are about the diversity of a population at a location. And as we build all of these studies, so this journal was conservation genetics, and more papers are coming out with this kind of spatial sampling where we're generating all of this spatial data on diversity. And that's really important, because it allows us to start to quantify what's going on. There's been a global loss of genetic diversity. So this was a science paper from 2016 where they looked at mitochondrial gene sequences and found that genetic diversity in the tropics was much higher than in non-tropical regions, but also found that habitats affected by humans hold less genetic diversity than wilder regions, while also controlling for that latitudinal gradient. So we have, this was sort of the first thing that suggested that we probably have had widespread loss of genetic diversity in taxa globally as a result of human landscape change. More recently, another paper in Science in 2022 used genomic data to look at, to develop what they call the mutation area relationship. So shortly, you should have more mutations where you have in the population than you, the bigger an area it is. So essentially more diversity in bigger areas. And so if we understand that relationship between those two things, then we can actually predict the loss of diversity given the starting area size and its current and contemporary size. And they estimated that more than 10% of genetic diversity globally has already been lost in a wide variety of species. This was 20 species, but had really, really good genomic data. This gets more important because genetic diversity is now being much more recognized as key to actually preserving biodiversity. So the United Nations Global Biodiversity Framework and the Convention on Biological Diversity has explicitly a 2050 goal that the genetic diversity within populations of wild and domesticated species, is maintained, safeguarding their adaptive potential. So that last line there. And this, interestingly, the draft version said that 90% of genetic diversity within populations is maintained. And if that was the case, this study would suggest we've actually already exceeded that minimum amount we wanted to retain. So in the final version, we have lost that explicit percent. And as part of this goal, there is a target, which is to ensure, as we see here, that to maintain and restore genetic diversity within and between populations of native, wild, and domesticated species. So genetic diversity is sort of now globally recognized and highlighted within the Convention of Biological Diversity as key to our vision for preserving global biodiversity. Great, we have some words, but how do we do that? And how we do that really comes back to what we call essential biodiversity variables. So these have been around a while, even before that particular framework. And this is really a global system of harmonized observations that we need to inform decision-making and policy. And so these are the sort of two key factors. So this is all available through GeoBond, so the Global Earth Observatory. And really, they are systemic biodiversity observations in standard formats that are open and interoperable. And within the EVBs, there's one specific class for genetic diversity alone, and that's called genetic composition. And within this genetic composition class, we are looking at 4 different specific markers that we want to try and record globally for every species we possibly can to actually inform all of these questions about loss. So intraspecific genetic diversity, so variation among individuals, then genetic differentiation, so divergence between populations, effective population size, so this is a sort of somewhat technical idealized number of individuals within a population given the genetic diversity, and then the level of inbreeding in populations. So these are now listed as the 4 key variables. Still not getting quite specific enough in terms of a practical sense. So conveniently, Sean Hoban, who is very involved in all of this stuff, and again, I have nothing to do with this, but I'm very interested in it, went and created this really great paper, which gets really explicit about EVBs. So we have our 4 classes here of genetic diversity, genetic differentiation, inbreeding, and effective population size. And they actually get really explicit about the definition, as well as the examples, the metrics you would use, and how you interpret that. 2 key ones I'll be talking about is particularly this observed heterozygosity, which is the evenness of diversity. So how evenly distributed is genetic diversity across the landscape? So together, these bring together a lot of wonderful information. So there's this global driver for understanding and retaining genetic diversity. We now have our methods by which we are supposed to classify, but now we have to actually use that data in some way. And that's where this emerging field of conservation macrogenetics comes in. This is a paper from 2021 that really is sort of highlighting the future and overview of macrogenetics and talks about how we take these single species population genetic studies and then actually use them to either generate raw data or use raw data to generate our own diversity metrics, to test those broad theories on what drives genetic diversity, or you can compile those reported values, those reported EBV values across all of the taxa and then combine them in some meaningful way. So, right, we're great. Generate your metrics, put your data together, you've solved a problem. Unfortunately, we're still far too early in the process for that to be true. We have a million different questions. So can we combine data generated with different sequencing methods? So what happens when you have whole genome sequencing data to generate your observed heterozygosity and your dd-rad-seq data? Do those come up with the same answers? Because the answer is often no. What if you assemble all your genomic data in different ways? So let's say you are running the STAX pipeline and you accept an allele if you have 6 copies, but someone else only accepts an allele if you have 7 read copies. So how does that affect your diversity? What if you do different analytical approaches? So you get your data, you've assembled it, then what? You have all of these filters that you can do to your data, but where you throw away data before you make your final estimate, which you have to do some of those filters, but how do they affect those estimates? And how do they affect your comparability across taxa? And even just basically, how does your number of samples that you use affect your estimate? So these are all really big questions we have to resolve in order to be able to get estimates of these 4 genetic composition EVVs that actually are genuinely comparable across taxa. So I think the first shot across the bow of the needs to be able to do this kind of work was published by more people in Melbourne. So Thomas Schmidt down at Uni Melbourne, and this is a really good paper, which basically showed that SNP-based heterozygosity is worse than autosomal heterozygosity. And they defined this as SNP-based heterozygosity, so SNP single nucleotide polymorphisms, so they're locations in the genome where there's only 2 alleles, and then you focus only on those sites within the genome. So you reduce your data to sites with only 2 alleles, and then on each individual, you ask for that individual, how many heterozygous sites are there, divided by the total number of biallelic sites that you retained, or autosomal heterozygosity, which is retaining those biallelic sites, plus all the sites that are fixed, so non-variant, and then you divide that number of heterozygous sites by the number of biallelic plus fixed sites. And I'm being really specific about biallelic here because we throw away all sites that have 3 or 4 alleles. Human studies show they're not that common, but the more we look at animals that haven't gone through successive bottlenecks, like humanity has, suggests that actually it's more common than we think. So in this paper, they then tested the effect of sample size, rare alleles, missing data, population structure. So what I'm going to have you do is just look at this top row first. So in this top row, they did 2 things. They have 10 samples down to 2 samples, and they also got rid of this MAC as minor allele count. So they had to have at least 3 copies of an allele in your population. And so obviously at 2, you can't have anything, but what's really important here is as your sample size goes up, your heterozygosity goes down. And so this SNP-based heterozygosity, so all of that throwing away of all of the non-variant sites actually has an impact. And what happens when you add more individuals is your denominator in that equation goes up faster than your numerator. So what's happening is you add more individuals, you add more or you add more just sites that you recognize, but you add them at a much faster rate than you add your variable sites, because we have a lot more invariant sites than we have variant sites in our gene. So just constantly adding individuals actually drives down your diversity. It's the same thing if you keep your rare alleles, but the difference you want to see here is comparing these two, in which we actually see that keeping rare alleles means we just have very different heterozygosity estimates. So the fact that your sample number and your filtering can affect your heterozygosity estimates for a population already says that this is not great for generating globally comparable metrics. And that they suggested using this autosomal heterozygosity, which seemed robust to sample number. And the other part I'll just show you here is that they found that call rate filters. So if you have 10 individuals, but this particular locus only has 9, you only have data for 9 of your individuals, do you keep that or do you not keep that? And what they found was it actually has an effect. So you actually want to have much higher call rate filters. So their recommendations were these. I'm going to come back to this. So autosomal heterozygosity instead of SNP heterozygosity is really important. You should omit sites with missing data, and population should be analyzed in independent runs. So I'm going to do a bit of a switch and then come back. So I'm going to introduce you to our study system, which is terrestrial vertebrates impacted by the Black Summer bushfires. We all know the Black Summer bushfires. I lived in Canberra at the time. It was quite miserable and very sad. So 9 million hectares burned, human lives lost, thousands of homes lost, some huge number of animals killed, and was having a potentially catastrophic effect on already threatened species. And as a result of this, I ended up leading a set of assessments for the federal government, funded through the TESS program, the National Environmental Science Program, and funded by Bioplatforms Australia. And we did detailed reports on genetic diversity and genetic structure in species as relevant to conservation management. As part of this, there's a huge number of people involved. I may have been in the boss, but that's only because I'm bossy and everyone accepted that. This took a huge amount of work from a ton of people, so I want to just say straight up, I cannot take at all the credit for this whole thing. So we generated genomic data for over 30 species of threatened vertebrate, and we didn't necessarily just stick with species we thought that were listed species, but also species we thought were biologically likely to be impacted by the fires in some significant way, often because they had genetic structure that was previously unrecognized. And this report and the assessment has been used for recovery planning and updated conservation advices of many of species, including the new listing on the EPBC of many species. But we have a lot of data of co-distributed species, which makes it sort of a first step for being able to do a macrogenetic study across a huge number of taxa. So we really want to know, naively, my first question was, what areas of the landscape are the most genetically important? So we have our genetic data. It's actually an extraordinarily unusual situation to have landscape scale population genetic data across a ton of species in the same region. They're trying to do this in California through the conservation genetics program there, but just that kind of data it's just really not quite in existence yet. So what can we learn about the spatial distribution of genetic diversity? What does this mean for recovery? And the first thing we had to do was get estimates we thought were comparable. So this is a paper led by my PhD student, Jared, that just came out in March, in which we revisited the Schmidt paper and agreed with a bunch of things and found some different things as well. On Twitter, my favorite comment about this paper was, I love when people's titles mean you don't even need to read the paper, which was really, really nice. So estimates of heterozygosity from SNPs are context dependent and often wrong. So these recommendations often didn't work for us. So one thing we definitely agreed with them on was that SNP-based heterozygosity is impacted by sample number. So we used one of their data sets, again, very, let's say, royal we in many of these things. So this is one of the grasshoppers used in the Schmidt paper. And then we included two species, a eucalyptus, which has been declining, but appears to retain probably a lot of its pre-European diversity because it's a really old tree. And then Latoria rubella, which is a really widespread species that also in the Kimberley is insanely genetically diverse. So we actually had a really wide variety from like a threatened grasshopper to a long-lived tree to a hyperabundant species. As what you can see is as the sample number goes up, in all cases, the heterozygosity goes down, and these different bars with different opaqueness is the call rate. And so what you can see is that the call rate always strong call rate filters. So missing data not allowed. So no missing data pushed heterozygosity down. But the important thing across all of this is that as you increase your sample size, as you increase your call rate filters, populations don't change the same. And what came out of this is largely that all of these things reduce the diversity in your most diverse population over your less diverse populations. So if you had 20 individuals of the Kimberley rubella and 20 individuals of your southeastern rubella, the loss in diversity by that high sample number and by that whole call rate are not the same. So it's not a proportional effect. And this is an aside here, but the SNP diversity in the tree frog actually doesn't even get the order of heterozygosity right, because this one, when you don't use SNP-based stuff, isn't even in the right order. So we completely agree, absolutely, SNP-based heterozygosity, you have to be extraordinarily careful. One thing they did find was that autosomal heterozygosity, this other method of calculating it, observed heterozygosity was robust to sample size. And when we used their data to do that, that was absolutely true. For this endangered grasshopper, autosomal heterozygosity, as you added samples, didn't really go down, not in any biologically meaningful way. But that was absolutely not true for our tree and our tree frog. So I understand, and we were able to utterly replicate what they did and why they came to that conclusion, but it was not correct for other cases. And so you can see we still have that sample number effect. And again, our problem is the slope of the decrease with increased sample number is not the same across populations, and the slope always appears to be somewhat higher for the more diverse populations. So if in this grasshopper you had 20 and 5 individuals across 2 different populations, you would get comparable autosomal heterozygosity estimates. But if you did that with Latoria rubella and you compared 20 to 5, you would say these populations are the same in terms of their heterozygosity, but they are absolutely not. And so when you look at this, so this is the effective sample number. And this bar is essentially the mean of our most heterozygous population. And these are the means of other populations at a certain sample number. And when you keep your sample number low, you actually get your proportion. So this is really looking at, do you get your ranking of diversity in populations correct at different sample numbers? So if you keep them small in 5, you can see 3 very similar and 1 less. That is actually very much the same. And with the Eucalyptus microcarpa, again, much the same, and also appears to be correct for the tree frog. But if you use every sample that's available, it does something really weird in the tree, and we're not entirely sure why, but if you use 40 individuals, this Western Victorian goes down way faster than everybody else. And really interestingly, if you use maximum available samples, so every sample we had, and it wasn't equally sampled across the taxa, somehow our Southeast population comes out as a much more diverse population than our Kimberley population. So unequal sample numbers can give you actually not even the wrong number for heterozygosity, but the wrong ranking of your species. And we also again found, they said, use a high call rate. And again, in that grasshopper, that makes sense. There was very little effect of this call rate filter on the grasshopper. But again, we had really big effects in the tree and the frog. So we don't agree that you should just use a high call rate. And in fact, we've found more consistency across all taxa at a lower call rate filter, so allowing more missing data when doing these estimates. So our amended recommendations share very many of those from the Schmidt paper, which was to use autosomal heterozygosity, keep your rare alleles. We doubled down and say you have to analyze your populations independently. You have to have constant sample sizes, but we add that your sample sizes actually need to be pretty small. And you should just do replicates if you have more individuals, so bootstrapping that. And you really need to test the impact of your call rate filters, especially if you have really diverse species or if you have populations within a species that are really differently diverse. So just as an example related to the bushfires, so these are Phylloria frogs. Phylloria or mountain frogs are arguably the most bushfire impacted group. If not the most, one of the worst. So essentially, if you wanted to make a little like tick chart of what makes you endangered, you live on top of a rainforest mountaintop in stream heads and be terrestrial breeding and very small. These guys are already all of those things. They're short range endemics and they were endangered before the fires. And fieldwork by people like Dave Newell and Michael Money have gone out, and it looks like the fires have had a significant effect, not just on population size, but the fires opened the forest to pigs, which, to quote Dave, might be the death knell for some of these species, is pigs going in and digging up their habitats. So for a couple of these species, Dave is establishing captive breeding populations. And so even just the population genetics, not even thinking macrogenetics, is really vital for setting up quality populations. And so when we look at autosomal heterozygosity versus SNP-based heterozygosity in these two scenarios, I just want to point out one key thing here. This is Sphagnum, which is actually the most widely distributed Phylloria. And when we use autosomal heterozygosity, we see that one of the populations, population 1, has about 4 times as much diversity as the next most diverse population. But when we use SNP-based heterozygosity, even at equal sample numbers, what we see is population 2 only actually has about 50% of the diversity. And so what's happening is we are using that wrong sort of method to get your heterozygosity is driving down the diversity of your most diverse population, and that will affect your plan for establishing a captive population and the recommendations of sourcing individuals. And even within autosomal heterozygosity, so that last slide was about how you would do management of the species, but if you're thinking about macrogenetics, if you actually filter all your minor alleles, what you see is these values of observed heterozygosity are reduced across the board. And if we're wanting to build studies that take heterozygosity estimates across species and put them across the landscape, we can't have really inconsistent filtering methodologies that in some species drive down our estimates, and in other species we don't, because then we just can't get comparable estimates. So filtering methods have significant implications for macrogenetic studies, but you also need to be careful about them for your PopGen too when you're doing conservation. So let's go back to the bushfires. So what areas are most important based on the distribution of diversity? So the question posed to me by Craig Moritz, who I was working for at the time, was like, what areas have the most diversity? And that's turned out to be a very difficult question. We have to ask a ton of questions. What are our metrics of diversity? Okay, we're going to stick with EBV type values, but how do we stack species in a meaningful way? Do taxonomic groups differ in their diversity? Can we put them together? Do we have to keep them apart? Do threatened and non-threatened species differ, and how would that affect our recommendations? So really, in practical terms, the question is, how do we go about using EBVs to understand that spatial dimension? And so again, royal we, I just put this in as me to my PhD student, Jared, go do this. And he has been doing a phenomenal job of that. So I am going to share some of the initial results, but they are very initial because the more we poke at them, the more questions we have. And it's really I think one of the first ones to really try and build this macrogenetic study. So first result, I think I'm quite happy with this result. It's something I've done before, so I'm quite familiar with, was just calculating phylogenetic endemism across our target taxa. So phylogenetic endemism is asking, where are the areas where unique range restricted lineages are held? So what areas have really distinct phylogenetic lineages, but that lineage is also only in a small area? So in doing that, we built phylogenetic trees using the data, divided that by the lineage distribution using this lineage range estimation model, and then average that across all the taxa, and this is the result. And what it clearly shows is a couple things in terms of our bushfire related taxa. The border ranges is really important, and I think that probably comes as no significant surprise to anyone. Also Kangaroo Island, which we included because it was quite fire impacted and has actually turned out to have a bunch of species that weren't really recognized sitting on there, and not just bird subspecies, of which there's a million that I am questioning of, but the frogs are certainly very distinct, as well as up Croombitville Burren. But we even still see really important stuff around the Florida Peninsula, Blue Mountains, areas as key phylogenetic areas. So the first question we had to ask is, can you put data together? And so the question is, does observed heterozygosity vary across taxonomic groups? So this graph here is our observed heterozygosity for a conservation unit versus the number of sites. And what you don't want is, when you first eyeball this, what it looks like. So this looks like higher heterozygosity with fewer sites. And that slightly suggests we have a problem with our read depth. But actually, if we ignore everybody over here, for reasons I will explain in a moment, and actually just look at this bulk of data, what we want is kind of a straight line. And we actually get that if we look at our taxonomic groups. So amphibians have higher observed heterozygosity within individual conservation units on average than reptiles, than mammals. And when we actually look at who these guys are over here, these are our most endangered individuals, so or individual species and populations, or sort of conservation units. And so if we ignore the things that are doing extraordinarily badly and have a real biological reason to have low heterozygosity, then we actually see pretty linear relationships between observed heterozygosity and sites. So that kind of tells us we have a pretty good method for generating comparable estimates, but it also tells us that taxonomic groups have different resulting observed heterozygosities. So then Jared went and used those lineage range models and assigned that average observed heterozygosity to each lineage. So we have its model distribution and its observed heterozygosity from a set of population samples. And some populations are not represented due to no sample numbers. So they're present in the lineage model, but they're not present in the genetic estimations. So this is just an example of one species, Asa darlingtoni, which is the hip pocket frog. And we kind of wanted to ask the first question, this is all very new, we're just trying to figure out the right approach to this, is even within a taxonomic group, so frogs are our most highly sampled group of organisms, so can we just chuck them all together? What happens? So it's 21 species of frogs and about 36-ish conservation units. And when we look at observed heterozygosity, what we're seeing is very, very high down around Victoria. And again, a little bit of a peak around the border ranges. But when we sat down thinking about this, okay, you know, that's like the first thing you would do with EBVs is grab all these data and just plot them. Great, okay, but we actually have threatened and non-threatened species in this data set. And if threatened species are lower than our non-threatened species, then what are we mapping? So we're mapping the distribution of diversity with true numbers that we think are real numbers, but in terms of prioritization, what does that mean? So maybe we need to not only split out our taxa, but we need to split out our threatened and non-threatened species, so our threatened species are not hidden and not prioritized. So if we separate them out by threat status, what we see is actually, you know, our not threatened frogs really were driving that previous map. That map really doesn't change much at all. Again, much higher diversity down around Melbourne region, which is quite interesting. And in our threatened frog species, it's interesting but variable. So our highest is the species in the border ranges. And again, like that first study I showed about genetic diversity, getting higher diversity as it gets more tropical in our threatened species, but not our non-threatened species, which is interesting. But still a little hard to interpret exactly what we want to do. And what we did realize is, well, we're still using raw values. So those raw values are real, but different species have different values. And we don't want to, if we want to weight every species equally, then we need to do some kind of weighting. So each individual contributes equally to the final map. And when you do that, what happens is you see kind of a leveling out of these values in the non-threatened species. And that's because a couple of really diverse species down here have other groups, and those groups get a bit more weight in other areas, but it didn't change this at all. And the reason for that, it turned out, was spatial scale. So spatial scale is another thing we have to deal with and figure out what's appropriate to the question. So we're having a discussion about the right spatial scale. And I pointed out that in the Phyloria, there are 2 conservation units split by an unsuitable valley. And that means that our conservation units are only 2 kilometers apart. So we start to get to this macrogenetic question of, like, within species diversity, can we even assess that? There's a lot of work to be done around all of these questions. So I feel like I was right on for time. So I guess my outcome to this, so keep looking at this space. We're making really good headway, I think, but practical macrogenetics is in its infancy. Outside of our need to develop published and really clear methods for reporting values for our essential biodiversity variables, and those not only need to be values, but reporting of the metadata of how they were generated, we also need to, we have so much work to do. So macrogenetics, I'm so hopeful, it's so interesting. We're gonna have so much we can do, but there are an immense amount of questions on how to make it practical. How do you practically do this to generate useful information for policy and management, to evaluate the ongoing impacts of climate change and habitat loss on species? And so there's about 50 questions. I just put up 5 that occurred to me immediately as I was writing this talk of things we need to assess. But there's a huge amount of work to be done in this field. And if you go on sites like Bioplatforms Australia, the amount of genomic data available is enormous. There's so much capacity to do fantastic work, but we still have a lot of work on how we put those together. So thanks to especially my lab, so Jared, who presented a lot of work he's been doing today, Sheng Yao for his work on the Phylorias, Rhiannon with her Phascogales, and Rhiannon Shembry, who was a research assistant on the bushfires and did a lot of work through that. And then everyone else involved in the bushfire genetic project who are also involved in this lineage project. So that's what I have for you today. I'm happy to take questions.
I want to say
Okay, I didn't quite hear you there, but anyway, anybody else? Sue? Okay.
hello. Yes, that's on. Hi, Renee. Thank you. I really enjoyed your talk. I agree with you that one of the problems that we have now is we have so much data and so many people want to do these global analyses, and they're integrating, they're data mining essentially, but there's problems with doing that, and it's not very, they're not being very thoughtful. And often the landscape approach is by far the strongest approach because it does take into account the context of what you're looking at. But I'm an ecologist, but I don't work with genetics. And I just was really curious. Obviously, as an ecologist, we always want to have more sampling. We always believe if the more you sample, the more of the variability within a population or within a community you get. So this idea that the more you sample, the lower the heterozygosity, I don't understand. I'm sorry if you already explained this, but could you do it again? Thank you.
Yeah, so it's kind of an artifact of the existing population genetic pipelines that we would like to see resolved. So let's say you've got 5 individuals, so you stack them together and you have a certain number of invariant sites and you have a certain number of variant sites. As you add more individuals, what happens is that breadth of invariant sites actually grows faster. So as you're adding more data, you add more loci in. As you add more individuals, you find more loci. But our read fragments are usually 68 base pairs long that we're putting into these pipelines, and they usually have 1 variant site. So to gain 1 variant site in the numerator, you on average gain 67 sites in the denominator. And so your number keeps going down and down and down and down. So what we're not at right now is the ability to take all that data for a single individual and confidently call variant sites and invariant sites, which is what we want to do. But we don't have the bioinformatic platforms to do that at the moment. And
yeah, okay, great.
We can get there. If that makes sense, it's about how data is added and our inability to assess confidently individual level estimates of heterozygosity.
Great. Other questions? Renee, I had a question for you. This is a big picture question and it might make you squirm a little bit. Let's see how we go. We're living in a biodiversity crisis. You're working right at the forefront of that. You're sort of at the where the rubber meets the road, I guess they say, in terms of studying how species respond, how populations respond. Do you believe, and I'd love to hear your frank impression, do you believe we are entering a 6th mass extinction right now? Do you see that the losses would be comparable to past mass extinction events, or do you see this as something more subtle and qualitatively different? I'm not trying to put any words in your mouth. I just want to know your thoughts.
Look, I think the data is very supportive of that being the case now. You know, the declines, and then, you know, like I said, we were talking about Perth weather. We have this year, it's still summer here. It's crazy. And we have had since October half of the rainfall of the next lowest year on record. And we're seeing, you know, areas of forest dying, you know, and we have our, you know, peat swamps in the Southwest that have things like, you know, the sunset frog that's a 35 million year old distinct lineage how are those going to persist in increased drawing at the rate that they're drawing now? So I would say I'm a pessimist in all of this, to be honest. I think we're probably on that track.
Okay, thank you. Anybody else? Anybody online? Okay, well, Renee, I think you've given us quite a lot to think about, and it's great that you tied it in with catastrophic events like the Black Saturday fires, because that's obviously something everybody knows. I thought you gave an extremely clear and interesting talk, and really no rough spots at all. It was a really good first-rate talk. So I learned a lot. And yeah, on behalf of everybody, thanks very much, Renee, and come visit us in Cairns next time you guys get in this area.
Thank you very much.
Thank you.